Today, I'd like to discuss actually these three topics. And the main topics is actually, upon the request, computational algorithms, which introduced in a program Genesis, and how to achieve high performance computing and how to accelerate MD simulations using supercomputers. But before that, I'd like to convince, in particular, the non-expert of MD simulations, why MD is important in biological science. That is the first topic. And the last topic, and so up to the first and second topic, I mainly focus on atomistic MD simulation, but we fully recognizing that multi-scale approaches are very important in biology. So in the last topic, I briefly introduced our multi-scale approaches. Okay, so that is the plan today. So this slide, so I'd like to show protein structure, dynamics and function relationship. So the movies show the conformational change of adenylated kinase, one of the famous enzyme which catalyze this reaction, ATP and AMP, from ATP and AMP to ADP. So to catalyze this reaction, this enzyme is working. And so crystal structures, we know that two distinct forms structures. One is open state, and another one is closed state. And closed state contains the ligand. So this means that this protein should move from open state to closed state. But without performing MD simulation, we cannot see intermediate structures. So that is one of the clear motivation for us. And also the difficulty for MD simulation is actually very large time gap of various motions of proteins. So you can see from this figure that the very first quick motion, that picosecond vibration, and also very slow motion involving not only one protein, but also many other proteins. And also folding, unfolding, or the protein-protein interaction takes a lot of time. So, but in both motion, even fast motion, we need to, if we want to predict accurately the quick motion, we need to investigate the harmonicity, which is less difficult than the just a harmonic approximation. And also for treating slow motion, we have to simulate the structure which is not resolved by experiment. So such multiple timescale nature makes the situation more complicated. But the theoretical computational approach is quite simple. Just the we are solving classical Newtonian equation motion. But the so in high school, probably both in Germany, you should learn this very simple equation in high school, Japan as well. And but in a simulation, we don't use this equation directly, but like this, using very small time step and integrate it and do the iteration so many times to simulate slow motion. And typically, we are using classical force field, that is energy function, and basically developed by three famous people who received the Nobel Prize in Chemistry 2013. And so theory itself is quite simple. However, we believe that it's quite useful to understand biological phenomena. So I'd like to show two examples which are actually related to our work. The first one is a recent hot topic in biology. So yeah, probably you may agree that is SARS-CoV-2. And in particular, I'd like to focus on spike protein on the surface of SARS-CoV-2 virus. So probably as you may know, this SARS-CoV-2 virus has a spike protein on the surface, and there are many spike proteins. And they are actually very important for the initial entry, virus entry to human cell. So the human receptor can bind the spike protein of the active form. And then infection starts. So this is very important, essential initial step of the SARS-CoV-2 virus entry to the human cell. And thanks to the cryo-electron microscopy, we know that down structure and up structures. And up structure is active and down structure is inactive. Only up structure can bind to human receptor. So we'd like to understand the conformational change and also the glycan interaction with this protein. So this is further more details about the spike protein structures. This is trimeric structure. Trimeric means there are 3 chains. And also this protein on the surface, there are many glycans that is shown like this. And actually in one chain contain 20 glycans, and it actually affects the structure and dynamics. And also the glycan interaction is important for antibody neutralization. So we try to simulate this protein with glycan and also surrounding waters. So the result is simply summarized in these movies. So we just perform the simulation of spike protein from down cryo-EM structures and also up structures. So like this, we added flexible glycans and try to see which glycans are important to stabilize these each structures. So from these simulations, we know actually 3 glycans are very important in either down form or up form. But unfortunately, by using cryo-EM electron microscopy, we cannot visualize such flexible glycan structures. So we perform MD simulation with glycan. So MD can add the additional information of this SARS-CoV-2. So this is one example. And second example is my calcium ion pump, which I have studied for a long time. And this protein is actually membrane protein, and this is a biological membrane. And this protein can transfer the calcium ion from cytoplasm to SR lumen. SR is one of the organella. And this protein is a very good target of MD simulation and also structural biology target. And there are so many different structures determined by X-ray crystallography. And by connecting the different X-ray structures, we can see kind of protein motion in this movie. However, this movie just connecting different structures probably may not so accurate. So we'd like to predict the more accurate dynamics in a biological membrane. So, and I actually studied this membrane protein from 2002 when I was working with Professor Kuroshima in the University of Tokyo. So he actually determined the structure for the first time, and he invited me to his laboratory to work with him. And when I was young, I simulated this protein using atomistic MD simulation. But I started this simulation from 2002, but at that time, computer was very slow and I can't simulate for a long time. So just I simulate 2 nanoseconds for this system. But even at that time, I treated lipid bilayers and also waters and protein in atomistic model. So the cost was at that time even very, very high. But anyway, by performing such simulation and with and without protonation states, protonation means if you look at these two figures, in one system, you can see this white bond. This is a proton attached in this amino acid, and another proton is attached here. But in different model, I don't add any proton in these models. So then I can see the difference of the stability of calcium binding site by these very short MD simulations. So this is very short. However, even at that time, we could fortunately publish this result in JACS in 2005. And after 16 years, we actually continued the calculation. And this year, we published a new simulation in PNAS. And in that case, we try to simulate the conformational change, very large conformational change, actually from one structure, E1P state, to another crystal structure, E2P state. So here, calcium ion is bound, but here, actually, calcium ion is released. So it's actually a big change. But to do this, just single MD simulation is not so efficient. So this takes, I think that in real time scale, millisecond was lower. So we cannot simulate directly just by MD simulation. So we are using kind of a replica-based simulation. This means that we are performing multiple image of the system. And here we just show only 4 images, 1, 2, 3, 4. However, in real simulation, this person, Chigusa, simulates 64 replicas, 64 images in between from the E1P and E2P states. And in the algorithms called string method, so she can optimize the images, and like from the straight line of the pathway and relaxing the pathway eventually, and finally physically meaningful pathways obtained. But to do this, we have to perform each replica longer than 100 nanoseconds. And after that, after getting the physically meaningful pathways, we can simulate umbrella sampling to get the free energy difference. It's actually a big calculation even for us. And we connected this simulation trajectory and getting the movies for describing the conformational change. Now the conformation is from changing from E1P to E2P. It go back to the original state. So eventually, this ADP is unstabilized, and calcium ion is here. And from the other side, now gate is open and water is penetrated. So that is the motion of the calcium ion pump, which we simulated. And again, so by performing this calculation, so we can obtain kind of free energy changes from one state to another state. And also, we performed different protonation states. I mean, in one simulation, we add 3 protons, and another one is just 1 proton. So this simulation suggests that 1 proton simulation is quite unstable. But if we add more proton in the E2P state, this becomes much more stable. So this means that with calcium ion, so this is the stable basin, and upon the conformational change, it becomes unstable, but probably calcium and proton exchange happens, and it go back to the another stable structures. So that is what we learned from these extensive simulations. So these two simulation is just example, and I don't have much time to talk about the detail of the biology. But what I'd like to tell, in particular from the second example, that is actually computational time. So in my first calculation of the sucker pump bias, we just performed 4 nanoseconds in total. So nowadays it's very, very short, but at that time, we seriously performed the simulation using nice clusters, but we could simulate such very short MD calculation with atomistic model. But now, in the latest calculation, we perform 16 microseconds. So you can see that the after 16 years, we can perform actually 4,000 times longer MD simulation. So that is, at least for me, amazing. And of course, after 16 years, computer everywhere become faster, but still 4,000 times longer simulation for the same system is, I think, great. And so hopefully after next 16 years, we can perform another 4,000 times longer. Then, we can fully simulate probably whole the cycle of this enzyme. Okay. So then, the first and second example suggest that importance of the computation in molecular dynamic simulation of biological systems. So then I switched to more computational side of our research using supercomputers. So this graph is probably seems to be familiar with many computer scientists. And this is top 5,000 taken showed last year. And this is this yellow line showed the top supercomputers performance increase. And this blue line is, I think, probably 500. Supercomputers. But anyway, I think as far as I know, from 2000 to now, Japanese supercomputer became top 3 times. The first one is Earth simulators in 2002 and 2004. And second time is actually K-computer in RIKEN in 2011, and just for very short time. And now Fugaku supercomputer, my background computer, is the top of the world. And fortunately, in this month, new top 500 was reported. And again, so this year, Fugaku is the fastest in the world. And this busy slide actually describes our own research group history and also supercomputer development from 2007 to now. And it was probably very lucky for me and for members to participate in such supercomputer development in Japan. And I actually joined in RIKEN from 2007, and this is almost the same as the start of the development of K-computers. And actually, in Japan, the supercomputer project, the flagship computers, we have both hardware project and also application project. And I participate mainly in the application project from the beginning. And the K computer starts its operation in 2012. And in parallel, we have application project, strategic research. And K computer finished its operation 2019, I think. But the much earlier, actually from 2014, we already started the hardware project for Fugaku computer. At that time, we just call it post K, but finally the name became Fugaku. And in parallel to this hardware project, we have application project priority issue, and now we have another 5 years. This is 3 years application project. And Fugaku started its operation last year, and we are fully using these computers. So, and in particular for using K computers, in our team, we started the development of MD software Genesis. And today I mainly discuss about how we struggle to develop this software and how we can achieve high performance computations. So we started the development of this software from 2009. Of course, that time K computer was not available, but we have had to prepare our own high performance supercomputers softwares. And then we continued the development, but for the first few years it was really hard time for us because program was not completed, but hardware development was finished. So we worked so hard and to complete our MD software. So today I would like to discuss our development of Genesis in terms of 3 efforts. So the first one is, so today I call it phase 1. That is Genesis version 1. And this is basically for K computer and GPU clusters. And second phase is actually more recent effort. That is Genesis 2.0 for Fugaku supercomputers. And the last one is like Replica-Exchange. We are developing and introducing many enhanced sampling methods for free energy calculations in Genesis. And because of these efforts, we could achieve several application studies like protein crowding simulations and spike proteins and the sucker pump I explained, and also protein drug binding studies. So before starting our development, I'd like to explain the computational bottleneck in energy and the force calculation of MD simulation. So this is the same equation and bonded terms and non-bonded terms in that is used in molecular dynamics simulation. But the most time-consuming part is actually non-bonded interaction, because bonded interaction, the computational cost is in parallel to the number of particles. But non-bonded interaction, without any approximation, its cost is order n squared. So this is a very expensive calculation for us. But in reality, we are using, in most cases, a particle mesh Ewald approximation. And in that case, we use we compute real space computation, that is somehow the order n, and reciprocal space calculation containing FFT calculation. So that part we call reciprocal space calculation. This is actually more expensive n log n operation. But anyway, we have to optimize these two non-bonded interaction in MD calculation. So I'd like to explain our effort briefly one by one. So the first thing we have to do is how to use a single CPU efficiently. And for this, we are using lookup table for the non-bonded real space calculation. And this is actually a very common approach in all MD calculation. But what is different between other approach and our software is we are using not just linear R squared table, but we are using inverse R squared tables, because this case is actually better in several points. So for example, this is a typical Lennard-Jones potential, and there is deep minimum. But if you use just linear distance squared lookup table, you have to put many points in this region. However, if you switch to inverse R squared, then this part will be much milder change. You can cover small number of points to approximate this function with lookup table. And in fact, if we compare the energy drift, that approximation, and also computational time, this inverse R squared is much better. So in terms of the accuracy and speed. And also computationally, this is also quite nice, because as I mentioned, that we can cover these region with small number of table section. And if we check in a real snapshot, so we can cover the most important interaction region with very small number of table sections. This means that in computation, cache miss is reduced compared to the just linear R squared table. So in this case, you have to put more than hundreds sections in this region. So you may suffer from many cache miss in the calculation. So this is one of our efforts to use each single CPU efficiently. The next attempt is how to use many CPUs efficiently. Then, so we developed a spatial decomposition scheme. So this is also quite standard, computational method in MD simulation, and probably in many other applications. So we divide the space into several domains like this, and then if you use MPI, each MPI process has responsibility on each domain, for example. So what we introduce in our MD program is midpoint cell method. So this is actually a cell version of original midpoint method, which was developed in D-Show research. So in the original method, the computation is done of the midpoint of two interacting particles. So for example, A particle and B particle interaction is computed at the midpoint cell. That is the second cell. And C and D, the midpoint cell is actually the number 4. And this interaction is computed here. So this is really good and low communication cost. However, you have to decide the which midpoint is used at each MD time step. But in our case, we developed several versions of this midpoint method. So we introduce concept of the cell inside of the domain. So now the division is more hierarchical. And the advantage of this method is the midpoint cell can be decided only once, just before the simulation, after you determine the division of the space, because the space is a space, so it not change during the MD calculation. So communication cost is much smaller. And also by this way, we can introduce hybrid parallelization. I mean that MPI parallelization and OpenMP thread parallelization, and both are naturally possible using the domain and cell division in the method. And the third computational trick we introduced is efficient three-dimensional FFT calculations. That is quite important in MD simulation because, as I mentioned, that we need real space calculation and reciprocal spare calculation and self-energy. Self-energy is actually constant, and you don't care. So problem is often called result from this reciprocal part because you have to compute 3D FFT. So I think computer scientists probably know much better than me, but this is quite quite intensive network communication, and we need to reduce the computational costs. And in our case, we actually have to perform twice because to get the potential energy values and also force from this reciprocal state to FFT is necessary in one time step. So what we have done is introduce volumetric decomposition into the 3D FFT. And this method actually increase the number of communication. However, in this case, much less communication amount is necessary in each communication. So in total, in particular for very large system, this method is quite efficient. And also, this volumetric decomposition is applied to reciprocal space. We can use the same decomposition to real space. So these two spaces share the decomposition. So we can reduce the communication gain. But in most cases, most of other MD calculation, real spaces decompose three-dimensionally, but the reciprocal space is decomposed in two-dimensionally, so communication is required. That is distinct features. So we applied this method into Fugaku K computers. And then we obtained very nice scaling for several systems. And using this scalable software, we try to simulate the cytoplasm of a very small bacterium, Mycoplasma genitalium, in collaboration with Michael Feig in Michigan State University. So he first made the model of this mycoplasma, cytoplasm, and we make the atomistic model together with Michael and perform MD simulation with K computer. And finally, we can get the trajectory and see the dynamics of the inside of the cell. And longer version of this movie is actually available on YouTube. So please use keyword RIKEN, bacterial cytoplasm, and MD. So yeah, this movie is actually gorgeous and nice, and with actually sound. So yeah, I hope you can enjoy this movie also later. And the Genesis is also available on parallel GPU computers. And in our implementation, we use both CPU and GPU. And communication intensive work is done actually on CPU, but the non-bonded real space calculation is done on GPU. And again, so we can achieve very good scalability, in particular on GPU part. And total performance is also quite scalable on GPU cluster Tsubame. So this is not the latest benchmark, but I think that even latest supercomputer with GPU, we can achieve very high scalable performance, in particular very large system. So that is our first phase. So, and the second phase actually for Fugaku supercomputers. And in this case, we participate in co-design for this new supercomputer. That is system development and application development should be cooperatively done. That is the concept of co-design. We participate in this co-design project from the beginning. That was fiscal year 2014. And eventually, we discussed and communicate with the designers of the computers and the developers. And what we found was the SIMD usage is quite important for the new supercomputers. And we try to examine all the expensive do loop calculations in the original algorithm in Genesis, and try to elongate the loop length as much as possible. And because of our efforts, so the loop length, originally it was very short, mostly less than 100. But after the modification of the code, we can extend it up to 1,000, 10 times longer. And because of this, we can achieve much efficient calculation on the new supercomputers. And it's not computational, but we also develop the much accurate kinetic energy evaluation in MD simulation. And it's actually a combination of the two conventional kinetic energy definition, the full time step and half time step kinetic energies. And we finally obtained this equation that is the most accurate and the temperature estimation. And by using this equation, we can extend delta T of the integration of MD simulation to up to the 3.5 femtosecond for short range forces and 7.0 femtosecond for long range force. And originally it was limited to 2.5 femtosecond. So we can get a lot of so acceleration by extending this delta T without losing accuracy of the integration. And then we can use Fugaku supercomputers. And in our benchmark, so again, we can perform very much high performance using a lot of computer node. And previously, our largest size was 100 million, but now we can achieve more than billion atom systems in computer simulation. So that is possible in the Genesis 2.0 and Fugaku supercomputers. So how much time can I talk?
If you could come to an end in a few minutes.
Okay, so I briefly summarize our effort of the enhanced sampling method and probably skip the third topic. So Replica-Exchange is a very old method, and you need to perform multiple images at different temperatures. And but recently we can keep the solvent temperature and just change the protein solute temperature. That is less to 2 or G rest. And using this kind of another parallelization, we can perform the protein ligand binding or drug design simulation more effectively. And yeah, so this method also available for 2D replica exchange, and you can find the bound states very efficiently. So yeah, in this way, so our Fringy approach is quite useful, not only the large scale simulation, but also biological simulation. And I have more slides, but probably I should stop here. So this is, recent achievement from our group, and that includes the crowding simulation and also conformational change, and the protein ligand binding and SARS-CoV-2 and heme transporter. This is also membrane protein simulations. So probably our group had quite a big benefit from K and Fugaku supercomputers, and also we are very happy to use such supercomputer for studying biological system in this way. So yeah, probably I had better stop here and welcome your question and comments. Thank you very much.